Yeah, it was good. Today got molecular genetics. Not gonna lie, I've been pushing through way too much content recently, and I'm low-key not retaining most of the new stuff, or I'm not really remembering the old stuff. So I just spend quite a bit of time just like understanding and memorizing the older stuff, and then fully understanding this as well, and just conveying things to long term. Otherwise, I'm just gonna get like cooked. You know what I'm saying? So, yeah, bro, life is so fried. Okay, what does semi-conservative mean? So it means like when DNA replicates, half of it will be from the original, and then half of it will be new. What type and where do bonds? So phosphodiester bond, we got the sugar phosphate. It's between the sugar and between the phosphate group. What direction does replication occur? It occurs from the 5' to 3' 5' to 3' direction. What two types of strand occur because of this? So because it only occurs in the 5' to 3' direction, the lagging strand will form Okazaki fragments. So yeah, just the two types of strand, leading and lagging strand. Because lagging, I think I'll get into it later, but it's like the RNA polymerase 3 has to keep on flipping back and just like restarting. So, anyways, four main steps to DNA replication. So we got initiation, we got elongation, or wait, so we got first we got the replication fork forms. After the replication fork, then we're gonna have like priming, like RNA, like primer will be put on. Then we're gonna have polymerase elongate it, and then we're gonna have termination. So what is the function of each of these, and then what would happen if each are not present? So helicase is responsible for unwinding um, the DNA. It'll like break the hydrogen bonds in between, and it'll just unwind it. So if it was not present, then DNA will not be able to unwind, and you're not going to have access to those nucleotides that you're trying to copy. Single-stranded binding proteins, those are responsible for holding apart the two separated strands. Otherwise, the hydrogen bonds will have like the two nucleotides will have an affinity for each other, and they'll try joining. So SSBs or single-stranded binding proteins will try and hold them apart. Topoisomerase, that's responsible for breaking and then reforming the DNA bond. I'm not fully understanding what bond is breaking and why it's reforming them again, but it's like because it's like coiled, um, topoisomerase, it'll just break and rejoin the DNA helix ahead of the bro. I'm just gonna check this out, bro. What does the total function? Relieving tension during replication and transcription. So it's kind of just cutting one or both strands backbone, allowing the strands to untwist and unwind, and then release them, resealing them. Okay, so it'll just unwind the DNA, and then it'll bind it together again when it's unwoven, kind of. So, yeah. So if it's not there, then tangles will happen, and like bad things could occur. Yeah. So tangling is the main thing. Primase is responsible for putting the RNA primer for where polymerase will bind. So it's just placing the RNA primer. Without it, then there will not be any starting point for polymerase to start elongating or start replication. Polymerase, so there's polymerase 3 and polymerase 2. The common thing between them is that they add nucleotides in the 5' to 3' direction. So polymerase 3 is responsible. I was incorrect with that. Polymerase 3 is responsible for adding nucleotides. They both add nucleotides, but polymerase 3 is responsible for like adding from RNA primer. It'll start from the primer and they'll just elongate. And then polymerase 1 is responsible for removing the RNA primer and replacing it with DNA nucleotides. So what's common between them is that they're both able to proofread themselves, so it removes or prevents errors. Ligase is just responsible for joining the Okazaki fragments. It'll just join the phosphate sugar backbone, forming a phosphodiester bond between them, and then it'll just make it look good as new. What are the key differences between the DNA polymerases? I think I kind of said that. Polymerase 3 is responsible for so what will happen without ligase? The Okazaki fragments will remain split, so you're going to just have non-continuous DNA. Polymerase, if you don't have that, then you just can't add DNA nucleotides. Like you're just cooked, right? So, what are the key differences between the DNA polymerases? Like I said, polymerase 3 is responsible just for adding nucleotides in the 5' to 3' direction from the replication fork, and then polymerase 1 is just responsible for removing the RNA primer and then just replacing it with DNA nucleotides. So yeah, let me see if I'm missing anything. Ligase, yeah, polymerase 1 removes RNA primer and replaces it with DNA nucleotides. Polymerase 3 replicates the DNA 5' to 3'. Okay, what does the lagging strand create? We create Okazaki fragments. Why does it happen? Wait, we don't even talk about that here. Why does that happen? Because so DNA replication can only happen from the 5' to 3' direction. The DNA strands are anti-parallel, so one will be from the 5' to 3', the other one will be 3' to 5'. And in order for, because you're going to have one replication fork, one polymerase will have to keep on resetting, placing RNA primer, like primase will have to place RNA primer, and then DNA polymerase starts, and then it waits until more of it's unzipped, and then they'll redo the entire process. What is the central dogma? Central dogma is just basically states that we go from a DNA, then we go to DNA just stores the information, then we go to mRNA, and then we go to proteins. So what is the process between DNA replication and mRNA? That's transcription, and then translation is from mRNA to proteins. What is the process of gene expression? It's just the process of kind of the central dogma, the process of going from DNA to proteins, because proteins are like the more reactive version. So it'll that's how you're able to express the change through transcription and translation. What is a gene? Gene is a specific sequence of nucleotides that code for a certain trait. I think we already talked about that. Specific set of sequence of nucleotide monomers. Yeah. What is transcription simply, and where does it occur in prokaryotes and eukaryotes? So transcription is just the process of going from DNA to mRNA. Going from DNA to mRNA. Where does it occur? In prokaryotes. It'll happen in a cytosol, because prokaryotes do not have a nucleus, and then eukaryotes, it'll happen in the nucleus, because that's where the DNA is. What enzyme is required for transcription? RNA polymerase. I would assume so. Yeah, RNA polymerase. What are the two types of strands in RNA replication? So we have the coding strand and the non-coding strand. The coding strand, what are the two other names for it? There are two other names, I just don't recall them. One's called antisense and one's called sense. Oh, a template and non-template. So the template strand is the non-coding strand, and that non-coding strand is also called the antisense strand. What is this? So the coding strand. So the template strand is what gets copied into RNA. So then in the end you're going to get the exact coding strand, except with uracils instead of thymines. So, okay. So the template strand. So the template strand is what is being templated, and then you're going to form a non-coding strand, because that's not what's getting coded. So you're going to have the coding strand and then template strand. So the template strand is non-coding, and that means it's antisense. And then we have the coding strand, which is the sense strand, and that is the coding. Okay. What does RNA polymerase bind to? What is contained within that region? So RNA polymerase in eukaryotes, it binds to the promoter region. It contains the Tata box. I don't think we go in depth in that. What is the relative position? How to describe? So there's upstream and downstream of a gene. So usually the promoter gene will be upstream or before the start of coding, mRNA and stuff. Um, yeah, the actual start site of RNA synthesis is downstream of the promoter, so it's after the promoter region. Okay, what are the 3 steps of transcription? So transcription, we're going to have initiation again, we're going to have elongation, and then we're going to have termination. Everything follows these 3 steps basically. So what happens in each step? So initiation, RNA polymerase will bind to the promoter region or the Tata box, and it'll just start replicating. How then elongation is just RNA polymerase will replicate, putting RNA nucleotides on the opposite strand, and then termination will stop with a stop codon. I don't remember what the stop codons are. I don't think we need to know that. So how does the body regulate which genes to express? What do they do? So they use transcription factors. This is in eukaryotes. Prokaryotes will use sigma factors. The body will use transcription factors. So they can either enhance or they could inhibit the RNA polymerase from attaching. So yeah, like transcription factors. I don't really know what else to say about that. Yeah, it doesn't say in these slides how the body regulates. I think that was just something that was said. But yeah, so it's like if the RNA polymerase is not able to attach to the promoter region, then the gene will not get transcribed. So yeah, what do they do? Okay, now I'm curious as to what I was looking for. Yeah, I think it's just gonna be promoter region. But why is it so slow, bro? What is transcription? Hello? Yeah, I'm gonna find that. Okay. How does termination occur? Stop codon. Don't really know what else to say about that one. That's pretty simple. Let me just make sure, just in case I'm being too confident. Okay, yeah, I was wrong. Wow. Stop codon is used for protein synthesis, bro. RNA polymerase will stop when it sees a AAAAA. Bro, damn, I'm actually dumb, bro. Okay, how does termination occur? It hits the AAAAA. What is mRNA processing? Does it occur in eukaryotes and prokaryotes? And what are 2 examples? So it only occurs in eukaryotes. It does not happen in prokaryotes. mRNA processing is just like post-transcription modifications to the mRNA that's created. 2 examples include the poly A tail, which is just adenines on the end of it, and then the 5' cap, which is a guanine, and it just allows it to attach to the ribosome easily. Yeah, okay, let me just make sure, because this is looking kind of rough. Process by which pre-mRNA is modified before translation into a protein product. So yeah, what does the 5' cap provide? Provides stability, and it provides an easier way to attach to the ribosome. What is it made of? It's made of a modified guanine, I believe. Yeah, GPPP. So yeah, guanine. So yeah, provides stability and it allows it to attach easily. Now, poly A tail, that's just a couple adenines at the end. Again, it helps with stability, and it I'm not sure what function it has. Probably just stability, and like giving like room for it to like degrade, probably. What process do the SNRNPs do? Yeah, so this is kind of cooked. I don't really recall what those are. snurps. What is the process called? I believe these are spliceosomes. So yeah, they splice. The process, they remove introns and they remove introns and join exons together. So it's called splicing. Um, and what does it stand for? Single nucleotide ribosome nucleotide phenyl. No, so I don't know. Small nuclear ribonucleoprotein particles. So yeah, these will just combine. What's the relationship between SNR and P? Yeah, so SNR and P, they they complex and they form spliceosomes. Like they just okay. It says sRNA and proteins form a spliceosome complex. This will help remove introns and then join exons together. What is alternative splicing and how does it allow? So alternative splicing allows you to reorder the mRNA that you splice. It'll allow you to reorder the exons that you join. So you could have different proteins for like you could have multiple proteins for different like you could choose which exons you join together compared to which introns. I mean which exons you join compared to like another one, and then you could have like multiple proteins from a single gene. What is the most effective way to prevent gene expression? You remove the promoter region. What are 2 ways to promote or prevent gene expression? So there you could like have gene enhancers or gene inhibitors. What is it? You could have activators or repressors. So activators will help transcription factors and polymerase to assemble, and then repressors will just not allow it to do it so easily. What is the most effective way? So the 2 methods of transcriptional regulation. So this is it's like MISI or something, MISI, mRNA, micro something, miRNA, microRNA, and then we have siRNA. So what is the difference between the two? miRNA. Okay, this one's I genuinely did not know this one. Some of these I'm just like I low-key forgot to like. I think I remember. This is mainly like grade 12, bro. 2 methods of transcriptional regulation. So there's MISI, microRNA, miRNA. That just inhibits translation. It's just like there and it's like a block, and it'll just like not allow RNA polymerase to continue through. Then there's siRNA, which is small interfering RNA. Well, I just like to think scissors because they just cut up the mRNA, like they just degrade it, and then you don't have any mRNA. So what are you gonna do? Like you can't do anything. What is the process of translation simply? So translation is basically just going from mRNA to proteins, protein synthesis, if you will. What does What does the genetic code allow? Bro, what am I asking, bro? What are we talking about, bro? What are we talking about? The genetic code is redundant and does not contain ambiguity. The genetic code allows for multiple. It's basically just a code on table, so it's like it allows you to have what is it called, like anti-codons. Yeah, bro, this material is kind of rough, bro. Like, okay, just need to sleep, bro. Then I'll be all good tomorrow. I need to like retain this information, bro. I'm explaining it very loosely, but maybe that's just saying that I don't fully understand everything. But I feel like in my head I understand what's going on. It all kind of makes sense. I just like when I speak it, it's like, bro, I'm just trying to connect the ideas. Not really like but I feel like I understand. Okay, anyways, genetic code. Like I feel like these are such niche questions that there's no point in like knowing this stuff, because it's breadth over depth, right? So, like knowing the exact qualities of the genetic code are not very useful. It's just kind of to help understand and grasp all the concepts that we learned today. So what was I saying now? Yeah, so genetic code is redundant and it's does not contain ambiguity. Yeah, so what is the star codon? What does the star codon for? It's methionine, MET. I don't know the name of it. It's just MET. I just remember that. What is a codon? It's just three mRNA nucleotides, and they code for a amino acid. What is tRNA for? tRNA, two main components are it contains the anticodon, the mRNA anticodon, so it'll have the opposite of the mRNA, and then it'll have that specific amino acid attached to it. What is tRNA for? It's just used for taking the appropriate it's used for taking the appropriate amino acid and then bring it to the ribosome, and then they'll join and form proteins. So what function do ribosomes do? Where can they be found? So they can be found on the ER or on the rough endoplasmic reticulum. What function do ribosomes do? Well, they just make proteins, bro. That's the main thing. That's their job. They make proteins, protein synthesis. Size of ribosomes in prokaryotes and eukaryotes. So eukaryotes are 60-40. Prokaryotes are... Wait, so eukaryotes are 60-40 combined to 80. And then prokaryotes are 50-30. They combine to 70. So, yeah. So what does amino-acyl tRNA synthase do? That's basically just adding the... it kind of like charges the tRNA. That's like, I think that's what the actual process is called. It charges the tRNA with the amino acid that it has, I believe. Let me just... I don't think that was something they talked about here. It's just something that I... Oh, for ribosomes, the main function, binding the tRNA anticodon with associated mRNA. Okay, well, yeah, prokaryote 50-30, 60-40, genetic code translation. Where are we at? Okay, so that's basically all. What is it? Let me just search this up. Amino-acyl tRNA synthesis. Yeah, it's just the attachment, the esterification. That's good to know, I guess. Anyways, continuing on, where are we now? We're at 9.5. We gotta get to... Oh my god, what is all this? I really don't understand that entire thing. Okay. How many binding sites are on the ribosome? There's 3 of them. There's the A site, the P site, and the E site. A, amino-acyl site. P, peptidal site, or peptide site. And then E, exit site. So, what does each site imply? So, acceptor site, amino-acyl site. That's where the amino acid will come in and join. That's where I'm just gonna bring it. But for the very first step, I'll talk about that initiation. But anyways, peptide site, that's where the peptide bond is held, like the growing chain, it's held there. Exit site, that's just where the tRNA exits. What happens during the 3 main steps of translation? So initiation, so the mRNA is going to attach to the ribosome. There's 2 different ways. I remember I learned about this in bio class, but it's like in one way you just like, there's like a ribosome and it's just like walking until it finds the star codon, and then the ribosome will attach to it. But what I think, okay, I don't fully remember this. There's like some step, but it was like initiation. But I actually want to know this one. So initiation, it's like the star codon will like initiate it. You know what I'm saying? So like MET, it'll be like, yo, like lock in, let's like start replicating, right? And then it's just like, yeah, I got you. Like we're gonna replicate. So can we load, please? Is this playing or not? Okay, so I think all we need to know is that the small ribosomal unit will attach the mRNA. Once it finds the mRNA unit for star codon, then the amino acid for the star codon will bind to that anticodon, and then with that, it'll signal the large subunit to come, and then we'll just continue on. Mind you, or keep in mind that the very first time that like the star codon, it'll start in the P site, otherwise it'll start in the A site, the amino acid site. Then elongation, we're just going to keep on going. Like, ribosome will shift every three codons, every three nucleotides or every one codon, and then a new amino acid will come in and it'll just fit in, and then, the ribosome will shift again. The P site will go to the E site, A site will go to the P site, the peptide bond will form in the P site, and then so on and so forth. How are we able to match up a lower number of tRNA to a high? Well, it's called wobble pairing, so the third nucleotide, or the third nucleotide of the codon, it doesn't have to be exact. The first two are probably usually good enough, so the very last one it allows for multiple coding. What are inteins and exteins? They're just introns and extrons on the protein level. So when you make the protein, you might want to cut out some amino acids here and there. And that's kind of what inteins and exteins are, I believe. Central dogma, like I said, it's like freaking going from DNA to it's going from DNA to mRNA to protein. Can it work backwards? Yes, in some cases they can, but usually not in human beings. Usually that's how viruses work. They'll do backwards transcription, and they'll form DNA from the mRNA that they have, and it's just not good. So, was the RNA world hypothesis? So, saying that RNA must have been the first genetic material to have risen. The two characteristics is that one, RNA is able, it's like reactive, like it's able to induce change, and the other one is able to store genetic information. So those are the best of both worlds. However, we have DNA because we don't want the best of, we want to have the best of one and the best of the other. We don't want to have an intermediate in between. RNA has two OH groups, so it's slightly reactive, more negatively charged, so it's like gonna be more reactive. So that's why I have DNA. DNA has only one OH group, so it's relatively more stable than RNA, so it won't react, and we don't have to risk the idea that like the RNA is just gonna, like the DNA is gonna react, or genetic material is gonna be like cooked. You know what I'm saying? So that's like the one reason. And then on the other side, reactivity. We do need reactivity. We need something to express and induce that change or the genetic material, and that's what proteins are for. Proteins are more effective at inducing change and stuff like that, or being reactive. So we just have proteins, DNA, and then we have RNA in the middle. But yeah, the world hypothesis basically just said, RNA was the best thing, and then we just started from there. What are telomeres? So telomeres are just end repeats of they're just like at the end of chromosomes. They prevent degradation because like they're not needed. So as you like repeat, like so. So telomeres, they're just like repeat ends at the end of chromosomes. Why do we have them? They prevent degradation. Why does shortening occur? So when we're replicating the chromosomes, right? When we have the RNA primer, it'll start like so close to the end, right? And we'll still have like okay, wait, let me think this through. So RNA primer is going to place the primase. Primase is going to place the RNA primer at the end of the chromosome, right? But like there's only a limited amount of space, so you can't exactly add all of it. Like, okay, wait, it's kind of common, bro. It's cooked, bro. I don't even know how to explain this shit. I kind of have a general idea in my mind. However, I must be incorrect in that I'm not able to explain it. Primers are removed from the very ends of the DNA can be filled in by DNA polymerase. But why not? So when we replicate in linear DNA, this is linear DNA, we're going to have primer at the end, but DNA polymerase can't bind to the very end, so we're going to end up replicating the DNA, but it'll still have like a small missing piece at the end. So that's how that's just how it freaking degrades, bro. Okay, it becomes shorter in the end. So um, that's like the main issue. Like that's how it degrades. So then, how are telomeres added? So telomerase, that's the enzyme. What it'll do is it'll act as a scaffold. It'll attach to the missing portion or the hanging indent or whatever of the repeated or copied DNA, and then it'll allow replicated like non-coding. It'll like attach the non-replicating DNA onto the strand, and it'll just be repeating. It'll just not be coding. So it's kind of like it'll like attach to like the broken part of it, like that's left over, and then it'll just extend that, and it'll just repeat that into like something pointless, like into like a non-coding. That's like not important. So yeah, telomerase carries RNA template. Telomerase binds to a 3' end of DNA, because remember we're going from 5' to 3'. So it's the DNA overhang. That's the word I was trying, not the not hanging indent. RNA template used to synthesize strand. Telomerase shifts to continue the process. Interesting. Okay, so now what is the Hayflick limit? So that's basically saying like, yo, what is the limit? Like, because like it depends on how long the telomere is. So it's like how many times can the cell replicate until actual valuable genetic information begins to degrade because the telomere is too short. So yeah, states that the number of possible cell divisions is dependent on the length of telomeres. Yeah, okay, so what are DNA mutations? So there's 3 points of DNA mutation. Yo, I ain't gonna lie, bro, we might have to just do part 2 tomorrow, bro. I'm not gonna lie. This is low-key mad work, bro. Tomorrow's Friday. I can't lie. I'm gonna review this material tomorrow and just, yeah, because I don't know any of this, bro. Yeah, okay, we're gonna start from 9.9 tomorrow.